Hi, my name is Shelby Carlson, and I'm presenting the poster on quantification of delta-9 tetrahydrocannabinolic acid among 16 cannabinoids in cannabis by liquid chromatography ultraviolet detection. The purpose of this poster is to create a liquid chromatography ultraviolet detection method for quantifying delta-9 tetrahydrocannabinolic acid among 16 cannabinoids in cannabis. The quantification was achieved using external calibration, standard calibration between 0.02 and 25 micrograms per milliliter. The measurement precision in triplicate was 5.1%. The method is not interfered by other cannabinoids present in cannabis. So basically, we're trying to be able to say like this is delta-9 THCA among other cannabinoids present in the sample. So breaking it down, starting with the introduction. So Cannabis sativa L is a cosmopolitan species that is widely distributed around the world. It's denoted by Cannabis sativa variation sativa and Cannabis sativa variation indica, which those are commonly referred as hemp and marijuana. Cannabis plants exert psychotropic effects when their delta-9 THC content approximates 1%. This is very common in marijuana, especially since it's more used as a psychotropic drug. During the 1950s, hemp became entangled with the U.S. efforts to regulate controlled substances, including marijuana. In the Federal Controlled Substance Act, CAS, of 1970, cannabis was defined as a Scheduled I substance. This law made medical and recreational cannabis use illegal. This kind of came to an end in December 2018 with the Farm Bill, which included provisions to facilitate the commercial cultivation, processing, and sale of hemp and hemp-derived products by excluding hemp from the statutory definition of cannabis, with a delta-9 THC concentration of not more than 0.3% on a dry weight basis. Cannabis products for medical purposes have been in a rapid growth. Cannabinoids are being explored as pharmaceutical targets with potential applications in the treatment of anorexia, pain, inflammation, multiple sclerosis, and other disorders. Delta-9 THC has found acceptable medical uses by the FDA to kind of help with nausea, vomiting, especially associated with chemotherapy. So a quantification of cannabinoids and cannabis products is important due to both legal and medically regulatory requirements. Recently, we have developed a LCUV separation of 16 cannabinoids, and then I will get further into the analysis of that. A little bit about the experimental, as you can kind of see on this screen, a zoom up of it. We first grind the cannabis sample using a Waring lab blender. Then we powder the cannabis sample using a Splex Genolite 1200, which just turns into a powder form. Then we use a 15 milliliter centrifuge tube, and we add 100 milligrams of the cannabis sample, the powdered version. Then we add methanol to make a 25 milligram per milliliter mixture. We ultrasonicate it for 20 minutes. After that, we centrifuge for 10 minutes, and then we filter with a 0.2 micrometer syringe filter, and then we do a serial dilution with methanol. Then below are the instrumental parameters for the LCUV instrument. Here are the structures of the 16 cannabinoids that we are looking at in this study. This first chromatogram is a LCUV chromatogram of the 16 cannabinoids, starting with CBDVA that elutes first, ending with CBT. And the elution pattern is due to the polarity with the column. So we are using a carbon 18 column with polar solvents for our mobile phase. This figure is an LCUV chromatogram of the 0.04 micrograms per milliliter of delta-9 THC concentration. As you can see, there's the peak for delta-9. And as we move through the chromatograms, it's just going to increase in concentration. So this is 0.04, 0.2, 1, 5, and then 25. This is the external calibration, standard calibration curve obtained from the UV chromatograms that I just showed you. As you can see, we have an R-squared value of 0.999, which shows that this is a really good line fit for our curve. On the x-axis is our concentration, and on the y-axis is response. So using the equation that we obtained from the curve, we can determine concentrations of delta 9 THCA when we know the response that we get from said peak. This is an LCUV chromatogram of a 50 microgram per milliliter cannabis extract and methanol. These are the cannabinoids that are present in the sample, and they're all labeled. And as you can see, there's delta 9 THCA right about a little before the 13-minute mark. Here is it again. All of our samples are performed in triplicate, so there's going to be three chromatograms for each sample. So the first chromatogram, this is the second one, and the third one. So in conclusion, a LCUV method has been developed for the quantification of delta 9 THCA among 16 cannabinoids in cannabis. The LOQ was 0.02 micrograms per milliliter methanol, or 0.04% delta 9 THCA in cannabis, since 50 micrograms per milliliter cannabis extract was analyzed. The delta 9 THCA content in a cannabis sample was measured to be 2.11% with relative standard deviation of 5.1% in triplicate. The method can assist the Drug Enforcement Administration, the DEA, to regulate cannabis products. I would like to end my poster presentation with a thank you for the financial support by the NIJ, National Institute of Justice, the National Science Foundation, and Western Illinois University Research Council. Thank you.